Hello, I'm so happy to be here. Today we're going to finalize the step 8 on how to lose weight compassionately, and that is to substitute simple sugars for complex carbohydrates. To do this, what we're going to do is we're going to integrate a lot of information that we have already covered so that we get a much bigger picture, a more holistic, whole body approach to understanding energy homeostasis within our body. We're going to see how all the tissues talk to each other and through hormones and other molecules, and how the brain then decides whether it's going to increase food intake and food expenditure, perhaps by increasing metabolism. Before we begin, let's have a look at what I have put together here on the board. Now, please don't get overwhelmed by all the big words. They are just words, and they usually just represent an area in particular within the brain. So, we have here in this square is the hypothalamus, and that is a part of the brain. Within the hypothalamus, you have the arcuate nucleus, and the arcuate nucleus, there's two populations of neurons. So each one of these is a group of neurons that plays a particular function. Then we have other populations of neurons. So when the green neurons are activated, it leads to satiety and increased energy expenditure. If the red pathway is activated, it will lead to hunger and decreased energy expenditure. Okay, we also have another part of the brain, the nucleus tractus solitarius, which also is involved in food intake and energy expenditure regulation, and that is part of the brainstem. Another part of the body that plays a role is the liver, and the liver has many GLUT2 transporters, so glucose transporters 2, when compared to the beta cells of the endocrine pancreas, which only has a few glucose 2 transporters, and this will become significant, and we'll talk about it later on. The endocrine pancreas, the endo, that refers to molecules going into the blood, into the circulation, so hormones, whereas the exocrine pancreas, which is another area within the pancreas, is responsible for moving molecules or substances into the exterior world, so outside the body. So in this case, it's going to be moving it into the small intestine, which is depicted by this square down here. Now, if you imagine there is a tube going from your mouth through your esophagus, through the stomach, the small intestine, the large intestines, and all the way to your anus. And so there's a tube, and inside that tube, that is part of the external world. And that's why exocrine means you're releasing stuff into the external world, so inside the small intestine. On top here, I have depicted the stomach by this square. Notice that both the stomach and the small intestine have an epithelium of cells that have different functions. If we were to zoom one of the small intestine cells, you can see that there are transporters which allow for the movement of glucose and fructose into the circulation. The other cell that I've included here is the gallbladder, which contains the bile that the liver has generated. Okay, I also wanted to say thank you to the beautiful child that wrote this on my whiteboard. So express yourself, love what you do. So I'm definitely taking that on board, and thank you so much. Okay. All right. So we're going to show the difference, the different effects that simple versus complex carbohydrate has on our body and the hormones and all that stuff. We're going to compare quinoa, which is a complex carbohydrate, with table sugar or sucrose, which is a simple sugar. We'll start off with quinoa, and we're ingesting the same amount in calories of quinoa as we do of sugar. So as you look at the quinoa in your plate, if you hadn't eaten for a while, you would be quite hungry and you would start to salivate. So this is the cephalic phase of digestion. So you haven't put any food in your body, but your body is already getting you ready to eat. So you will salivate, your vagus nerve will be activated, and you start secreting acid within your stomach. Hormones such as glucagon-like peptide 1 will start to be released into the circulation from Earl cells. So glucagon-like peptide 1. Glucagon-like peptide 1 plays a role in the release of insulin in preparation for the absorption of glucose into the blood. It also plays a role in activating the nucleus tractus solitarius, and it does this through the vagus nerve, but I've just drawn an arrow straight to it. The nucleus tractus solitarius plays a role in promoting satiety and a sense of fullness. Okay, so we haven't eaten the quinoa. We're gonna eat the quinoa now. We put it in our mouth. We're chewing to grind the food so it's sort of an appropriate size. It's mixing with the saliva. Within the saliva you have lipase, a lingual lipase, which breaks down fat, and you also have amylase, which helps to start breaking down the carbohydrate. So that food then travels through the esophagus into the stomach. In the center of the stomach, there's still going to continue to be digestion by the amylase and lipase, so that whereas because it hasn't been in contact with the hydrochloric acid from the stomach, however on the outside of the food there's some contact with the hydrochloric acid, and so the amylase in particular will become denatured. What does denatured mean? It means that the three-dimensional structure of the protein is destroyed by breaking the bonds, and so you get a linear form of the protein, the primary structure, which is just a string of amino acids. And this allows for proteases, enzymes that break protein, to come and break it up into smaller peptides or single amino acids. So quinoa has about 10 to 15% of protein. So there will be some protein in the stomach. It also has some fat. And it also has 60 to 70% carbohydrate in the form of starch and fiber, so mainly starch. So we've got carbohydrates. I just might put carbs. Just put carbs. Okay, so starch is made up only of glucose molecules, so there is no fructose in this case. If you want to know more about the different types of carbohydrates, look at the first video on step 8. Okay, so as the food is in the stomach, there's also cellulose, which is, let me just put the charger on there. Okay. So quinoa will also have cellulose, which is the carbohydrates we can't digest, that are fiber, and that's going to bulk up the food. Your stomach is going to expand a bit more. The expansion of the stomach is going to activate the vagus nerve, which then causes a feeling of fullness. All right, now we have protein will be detected by the G cells of the stomach, and this is going to result in the release of gastrin. Gastrin then acts on Nevi cells. So it acts on the parietal cells, and that causes an increase in the secretion of hydrochloric acid. So we had already increased the secretion of hydrochloric acid during the cephalic phase, which is before you eat. But now it's further enhanced through the action of the hormone gastrin. So we get an increase in HCl. It's also going to act on the chief cells of the stomach to cause the release of an inactive form of a protease. A protease is an enzyme that breaks down protein. So it acts on these chief cells, and you get the release of a molecule known as pepsinogen. Pepsinogen then through the hydrogen chloride gets broken down to the actual active enzyme, which is pepsin. Pepsin can then activate a lot of other pepsin molecules, so you get like an exponential effect. Okay, so some of the chyme, or the other thing that gastrin does is it causes an increase in motility further down in the gastrointestinal tract, so that you move the food that has already entered into the intestines further along, and so you can fit in the new food that has come in. It also causes contraction or increased motility within the stomach. Okay, we then get some food coming into the small intestine, some of the chyme. Now the food that comes in is going to be acidic because we've just secreted a whole bunch of hydrochloric acid. So there's going to be a decrease in pH. The decrease in pH is detected by the S cells of the small intestine. And the S cells then secrete secretin. Secretin is also known as nature's antacid. Secretin can go and inhibit gastric emptying. So that is, it slows down the movement of food from the stomach into the small intestine. Secretin also acts on the exocrine pancreas to counteract the acidity, the increase in hydrogen ions, by causing the release of sodium bicarbonate into the intestine. So we have sodium bicarbonate, and this basically neutralizes the acid chyme that comes in. And you want to do this because a lot of the enzymes require a more neutral pH in order to function effectively. The other thing that's going to be coming through are going to be proteins and fat. So proteins are small peptides, amino acids. So they're going to come across protein and fat. And these ones are going to be detected by the eye cells of the small intestines. The eye cells of the small intestine then release cholecystokinin. So cholecystokinin adds on to the effect that secretin has on gastric emptying. So there's a decrease in gastric emptying. It also acts like, similarly to glucagon-like peptide 1, it also acts to, actually up here, on the nucleus tractus solitaris to promote satiety. The other thing that CCK does, or cholecystokinin, is that it causes the contraction of the gallbladder. So CCK acts here, and you've got bile stored here. The bile is released into the small intestine, and it helps to emulsify the fat. The other thing that CCK does, it acts on the pancreas, the exocrine pancreas, and this is to release the enzymes that digest the protein, the carbohydrate, and the fat. So the amylase digests further the carbohydrate, the lipase digests the fat, and the proteases digest the protein to its amino acids and small peptides so that they can be absorbed and taken up by the body. Okay, and have we got it on? Then we got the carbohydrates. So the carbohydrates are detected by the L cells, and that further causes the release of glucagon-like peptide 1, so you get a second surge, and you will get more insulin released, as well as more. It will affect the nucleus tractus solitarius more. Okay, so we have now digested the carbohydrates so that they're disaccharides now. They're just 2 molecules of glucose, because remember we started with starch, and starch is only made from glucose. So we have maltose, and so maltose will be broken into monosaccharides or single glucose molecules by the enzyme called maltase. So you can see the 2 glucose molecules attached here to the enzyme, and it will release 2 of these, which can then be absorbed into the cell through the sodium glucose co-transporter. So what that means is that 1 sodium molecule goes in with each glucose molecule. The glucose will then leave the cell through a glucose transporter 2, so a GLUT2 transporter. It will then go into the interstitium and eventually go into the circulation, where it will travel to different parts of the body. So you will get an increase in glucose in your blood. So increased glucose, so I'm just putting G for glucose because it takes up less space. So the increase in glucose, it's going to go actually into the beta cells. And remember, it generates ATP, and that causes eventually the release of insulin into the circulation. So if you want to know exactly how that happens, look at the video on glucose from blood to energy to storage, which I've done before. Insulin then acts on the different tissues such as liver, fat and muscle to move the glucose into those cells to use for energy and to store it. Okay, insulin will also travel to the brain, and it will act on the arcuate nucleus in these neurons, the POMC and CART neurons. So POMC stands for pro-opiomelanocortin, and cocaine and amphetamine related transcript. These then activate the paraventricular nucleus and the ventral medial nucleus, which both result in satiety. There have been experiments where the ventral medial nucleus is destroyed in animals, and those animals become extremely hungry. On the other hand, if the lateral hypothalamic area, which has been associated with hunger, is activated, the animals become hungry as well, quite ferociously. So insulin is directly proportional. The insulin that is released is directly proportional to the amount of glucose that's gone into the beta cells. And okay, so most of the glucose will be stored as glycogen in the liver. Whatever's left over will be stored in fat cells, and it will also be stored in muscle as glycogen and perhaps a little bit of fat. Okay, now the important thing to note is that insulin also, if you remember, acts on fat cells to cause the release of leptin. Leptin is proportional to the amount of fat that you have in your body, and both insulin and leptin are letting the brain know what your energy status is. And so we'll compare, and that's important for when we compare it to fructose. The thing to note is that glucose is not gonna go up very high in a hurry, because you have fats and proteins and the pH all negatively feeding back to inhibit gastric emptying. And if you have food in your stomach, you're not going to release ghrelin. The most potent inhibitor of ghrelin release is believed to be glucose. insulin has also been shown to play a role in inhibiting ghrelin release as well. Fat and protein also inhibit ghrelin release, but it looks like it's not as much as glucose. So by having a complex carbohydrate, you're actually keeping the food in the stomach longer, therefore releasing less of the hunger hormone, and also you're releasing the glucose into the circulation more slowly, so you don't get insulin spikes. And so this is a much healthier way of introducing glucose into your cells. Now let's contrast this to having table sugar or sucrose. So you will have the cephalic phase. If you hadn't eaten and you're looking at a tablespoon of sugar, you might salivate as you're preparing your body to consume the food. The amylase, salivary amylase, is not going to be able to break the carbohydrate further because it's already in its disaccharide form. And the disaccharides actually get broken down by the brush border in the small intestine. So the amylase in this case is not going to be used. So inside the stomach, you're not going to have protein. If you don't have protein, which is the main stimulator of gastrin, there's not going to be that much gastrin release. If there's not that much gastrin release, there's not going to be as much hydrochloric acid release, and there's not going to be as much pepsin released. And there's no fat. There's no fat in sucrose. Sucrose is just fructose and glucose together. Sucrose. As you put the sugar in your mouth, what is going to happen is that it's going to dissolve and it's going to be liquid. So there's no solid. You only when you're ingesting it, it's going to end up as a liquid, which means that it's not. It's pretty much going to go straight through to your small intestine. It's going to be very hard to stop it from proceeding through the small intestine, even though there's still going to be a small decrease in pH, which is going to cause a bit of secretin to be released, and a small decrease in gastric emptying. So sucrose, so the other thing is the vagus nerve is going to be activated if you're hungry and you see the spoon of sugar. So you are going to release some hydrochloric acid, which is from the cephalic phase. So that's still going to be there. You're also going to have the glucagon-like peptide 1 as well being released in preparation for the glucose entering the circulation. Okay, so there's going to be a little bit of a decrease in pH, so you're still going to get the release of a little bit of bicarbonate into the small intestine. The proteins are not going to be there though, however, and the fat. And I guess cholecystokinin is not really going to be released. And cholecystokinin is fat is the most potent inhibitor of gastric emptying. So that's going to be absent. Also, the effects that CCK had on the nucleus tractus solitarius to make you feel full and satisfied, that's going to be gone as well. So there's less of a fullness sensation. Now, there's only going to be sucrose is made up of 50% glucose and 50% fructose. And so there's going to be half of the glucose present. And so you're going to end up with a lot less insulin because fructose doesn't cause as much of a release of insulin as does glucose. And the reason is because there's not many GLUT2 transporters, and so fructose can't easily get into the beta cells. Most of the fructose will actually go to be processed by the liver. If you want to know more about that, look at the video on fructose, should you or shouldn't you, to see the effects and how perhaps fructose might be promoting an increase in triglycerides or fat within the circulation and within the liver and the whole body. Okay, just before we talk a little bit more about the hormones, I just wanted to just quickly touch on how sucrose is digested. So it's already pretty much digested, but as it enters the small intestine, there's an enzyme found within the membrane of the cells called sucrase, and that will separate the fructose from the glucose. We already talked about how glucose is absorbed into the circulation. Fructose uses a different transporter. It uses the glucose 5 transporter, and then on the basolateral side, on the other side of the cell, it uses the glucose 2 transporter, and then eventually it will enter the circulation and mainly be metabolized by the liver. Okay, so let's have a look. What will be happening in the stomach? So the main inhibitor of ghrelin release is glucose. Now there's not going to be much glucose because the gastric emptying is going to be quite quick. So the person's going to start secreting ghrelin a lot quicker than if they had consumed a complex carbohydrate. There's going to be, proportionally to the consumption of calories, there's going to be a lot less insulin. So perhaps the brain may think that you actually haven't consumed as much as you have. There's less leptin that potentially is released because there's less insulin to stimulate the release of leptin. You don't have the CCK to further promote satiety. Fructose has been shown to have not as much effect on the inhibition of ghrelin. So overall, you can see that by consuming simple sugars, particularly those ones that have fructose in them, you're going to feel hungry quicker, and therefore you overall during the day you're going to consume more calories and potentially put on more weight. Another thing to touch on is that there are many studies on animals showing that consumption of fructose makes the animals hungrier, and that they have more of a reward towards those foods as well. And there is some of these similar information coming out in human research now. And so I guess the ghrelin, just to touch, remember, goes and acts on the red pathway to cause hunger, and insulin, leptin act on the green pathway to promote satiety. So having said this, when you're going to eat carbohydrates, perhaps look at the carbohydrate that you're eating and choose the complex carbohydrate over the simple carbohydrate. Next time we're going to look at the reward system, and this is not a step, it's more like a consolidation of information that's going to link the first video that I did on stress, as well as fructose, and how different foods are labeled and rewarded by the brain. Until then, have a great week.
And don't forget to like and subscribe down below.